Thank you very much for the introduction, Sai. And can you see my screen? I take it it's working.
Looks perfect. Thank you.
Excellent. Thank you. Thank you very much for the excellent talk, Teresa. This is going to be a bit of a change in scope. This is going to narrow into the work of a handful of academic labs at Karolinska, focusing on currently questions surrounding variants. So as Teresa mentioned, the variants were inevitable, but we did spend the first part of the pandemic sort of maybe convincing ourselves that the mutation rate was lower than something like influenza, and maybe being optimistic that variants wouldn't become a problem for quite some time, and that turned out to be quite wrong. This was sort of upended. This was the picture where everything in blue is the sort of pre-variant of concern, so pre-VOC sort of virus. Towards the end of 2020, we saw beta in sub-Saharan Africa, alpha emerging in Europe, and gamma in South America. And these swept through in a matter of months to sort of dominate the global picture. And you can see there's delta emerging here, and in the middle of March 2021. And another 4.5 months later, Delta's overtaken, almost the entire globe, except for a holdout sort of in South America. If you push this forward as close as we can get now with the current sequencing data, it looks like Delta's swept all the way through. So it definitely has some strong advantage. So clearly variants are a major concern. They're of large consequences for at least the sort of spread of the virus, given how quickly they can overturn the population. But we have great vaccines, as you've heard a lot about in the previous presentation, and these vaccines elicit varying degrees of neutralizing antibodies, and even given all the caveats Teresa mentioned about how you define endpoints for vaccine efficacy, it really does seem like the potency of the neutralizing antibody response is a great predictor of the efficacy of the vaccine. And when you see a loss of neutralization against a variant, that does seem to go hand in hand with a drop in vaccine efficacy. So the alpha variant is only mildly in immune escape. It doesn't have a strong constellation of immune escape mutations, and you see a correspondingly mild loss of vaccine efficacy. But other variants have shown much more dramatic changes in efficacy when they're larger, when they have more consequential escape mutations. So just to go into the details about that a little bit, this is the famous spike protein that Teresa mentioned multiple times. And on that spike protein, there are two major targets of neutralizing antibodies. The foremost one is this receptor binding domain, which is, it's at the top of the spike protein, and it engages the ACE2 host receptor for entry into cells. And if you look at the sort of union of mutations from all of the variants of concern on the receptor binding domain, they're very concentrated in close proximity to this RBD ACE2 interface. So they're either in the interface or they're sort of adjacent to it. And that makes sense because antibodies that bind near that interface are going to be blocking ACE2. So, something that is a large, mutations here could be expected to sort of kick off a lot of those antibodies and interfere with their ability to block ACE2. And then a secondary specificity is this N-terminal domain on the sort of side of the spike protein. And the mutations there look a little different. The NTD has these protruding loops. And so this is an example from Lambda, where you see a large deletion of a number of amino acids that reshapes how this loop looks. And that's quite reminiscent of immune escape in early HIV infections, where you see sort of insertions or deletions that change the conformation of something. And again, that would kick off antibodies that bound to that target. But it's really important to bear in mind that mutations aren't just inducing immune escape. There are mutations that are of consequence for other reasons. So you can have mutations in the ACE2 interface, so like this at this position 501, that's enhanced affinity for human ACE2. And these are expected when you get a virus that's just crossed the sort of species barrier and is now circulating in a new host. And so not all of the mutations in the ACE2 interface are going to be of consequence for immune evasion. And then there are other mutations that affect the sort of behavior of the spike more macroscopically, and some near this cleavage site that's important for the sort of movement of the spike and entry. And those have consequences that are completely unrelated to immune evasion. And you can kind of think about these variants along the sort of two axes. The one is, how aggressively do they spread? And the other is, what is their fold reduction in antibody, antibody susceptibility? This is sort of my approximation of that picture. And you can see the Delta variant is not very high up on the immune evasion axis, but it's extremely far to the right on the transmissibility axis. Whereas something like mu is also very transmissible, but not as transmissible as Delta, but extremely sort of worrying in terms of the immune evasion axis. So it's almost like Delta is currently preventing mu from circulating, which might actually be a fortunate consequence of how things happen to play out. But basically, given this picture, we were involved in sort of preclinical immunological studies, and one of the questions that we wanted to ask, once we saw the beta variant emerge, is can we boost antibodies against these variants? So I'll talk about two little studies that we've performed in that sense, and the one is a small pilot study where we had some animals that we'd already immunized with the original, founder spike variants, sort of 2 doses, 1 month apart, and their antibodies nicely recapitulated what we see in vaccinated humans. They had very potent antibody responses that neutralized the founder variant very well, but they had this sort of 9-fold reduced neutralization against the beta variant. So that was a nice model for asking, can we boost them? And we had immunized those animals sort of 6 months before running this study. So it was just fortunate that the timing mapped onto, a nice model for a live vaccine human setting. And we asked, can you boost responses against variants using just the receptor binding domain immunogen? So it's not very immunogenic by itself, but we kind of thought that the requirements for boosting immune responses, even if you're trying to boost them against variants, might be lower than those for initiating new immune responses. And so this is the question we asked. And briefly, basically, the results from that are very promising. So this is the neutralizing antibody trajectory that we see in these animals. So you can basically see after 1 dose, you see almost no antibodies. That's sort of expected. After the second dose of this is the founder spike protein, you see very potent responses against the founder variant, reduced responses against beta, and these sort of decay over time. And then after boosting with this beta variant receptor binding domain, we get very potent antibody responses at similar levels against both variants. So, and this was consistent across all the animals. And then asking, was this a specific response to beta, or did this boost things across all the variants? It turns out it seems to be that you get these heterologous responses where they're boosting across all the variants. So where the serum after the founder spike immunization was potent against founder and alpha, and then had varying loss against gamma, delta, and beta, they were relatively flat across the board and very potent after this variant receptor binding domain boost. And we even boosted responses against SARS-CoV-1, which was actually quite unexpected and pleasant. And this is just the picture of the immunogen. This is the receptor binding domain with the 3 beta mutations on it. So that's what we're using as our immunogen in these animals. But we wanted to ask, because we didn't actually know about the consequences of the variants in human populations yet, because when we were setting this up, it was quite early on. So we wanted to ask, do the differences we see in the neutralizing antibodies translate to differences in vivo protection? And so we set up a cross-species passive transfer experiment where you take serum from the immunized animals and you transfer it into transgenic mice that are susceptible to SARS-CoV-2 infection, and you challenge the mice. And this lets you, set up nice repeated designs, and you can have more animals, and it's a convenient way of investigating these questions. And basically, the answer from this might be exactly what you expect. If you transfer serum after 2 doses of the founder spike protein to mice, those mice are fully protected from a challenge with a wild type isolate. So this is a spike without any of the VOC mutations, but they're only partially protected against beta challenge. So they don't exhibit as severe disease as completely unprotected mice that didn't receive any serum transfer, but they definitely show a weight drop, and they show enhanced, there's more viral load in their lungs, and they show a more, worse off lung histology. However, if you transfer serum from the immunized animals after this variant RBD immunization, those mice are protected from wild type challenge, and they're fully protected from challenge with the beta isolate. So you see no reduction in weight, and there's strong differences in the in all the other metrics. So that was one study looking at can we boost responses against these variants using a small, modified, convenient to work with immunogen. And I think the results are quite positive there. And, we're a small academic lab. And so what we're doing next is asking more detailed immunological questions about this. So you can dig in detail into the antibody repertoires for these responses and ask questions like, do the antibody lineages that are accounting for this response, are we boosting pre-existing lineages just in magnitude, or are we seeing mutations in the antibodies to adapt to the new variants, and ask all of those detailed questions. That's what we're doing now. And then we're also investigating more divergent antigens. And, do we see a similar behavior when the antigen is more divergent than just 3 mutations from the founder? And, might we expect these concerns about this phenomenon of original antigenic sin, where sometimes it can be challenging to boost different responses? Maybe those will become more of a limitation for more divergent variants, because they didn't seem to be a limitation for the current, at least, the beta variant of concern when we used that. So that's basically the sort of immunization work that we've been looking at, and in the context of these variants. And the other line of work we've been engaged in this series of projects is, can we find single domain antibodies or nanobodies? And these are potential therapeutics that neutralize variants of concern. So briefly, some background. This is a conventional antibody with that it's a Y-shaped molecule which has a paired heavy and light chain on it. And some sorts of animals have an alternative antibody format that doesn't have light chains, and it's basically missing this domain over here, and that means that it doesn't pair with light chains. And these can be worked with very conveniently. They can be studied in detail very conveniently, because you don't have to worry about the chain pairing, and they can also be functionalized in this nice small stable format. So those are called nanobodies. And so the system we work with is alpacas. So this is Tyson, the alpaca, who was the first animal immunized with this. Basically, this is in collaboration with the neighboring lab, especially Leo and Jerry. They're sort of next door to my office. And Tyson was the first animal we worked with. So briefly, Tyson was immunized with S1. It's sort of part of the spike, and it's not a great immunogen. We sort of figured out retrospectively. And then the blood sample from Tyson is reconstituted. You amplify out the entire nanobody repertoire from the animal, and you clone it into a phage display library. And then you can enrich the phages that bind, using this nanobody against whatever antigen you want. And then there's a traditional way of screening single colonies, but you can also, what we did is we added a next generation sequencing, it's like a deep sequencing arm that lets you look at the whole change in this phage library over the panning steps and analyze the enrichment. And actually the results from that for Tyson were very concordant. We found basically one working nanobody in Tyson, which we called Taiwan. And this was published relatively early posted early, published a little later in 2020. And Taiwan, as a monomeric nanobody, has what we would now say, low to moderate potency, but as a bivalent binding on an FC construct or in more multivalent arrangements is exceptionally potent. However, Taiwan shows, so Taiwan has some other nice properties. It binds threefold symmetric across the apex of the trimer, which means it doesn't self-compete. So I think that helps its neutralization profile a little. But Taiwan has no cross-reactivity against other variants at all. As soon as you have any mutations in that S2 interface, Taiwan is sort of kicked off and it's not cross-neutralizing. So after immunizing Tyson, we had improved the immunogens we were working with a little bit. And we immunized a second alpaca called Funny, who's far less photogenic. And we see a roughly 2 orders of magnitude improvement in the serum neutralization potency. So just overall, the antibodies in Funny are much better than they were in Tyson. And we spent some time mining these in detail. And we've also changed our screening strategies. So instead of doing a single screen against one antigen, what we're doing is panning in different ways. So we have enrichment against whole spike, we have enrichment against the receptor binding domain alone, and then we have enrichment against spike in competition with receptor binding domain. So all the RBD responses will get downregulated in that particular panning arm. And then we sequence all of these libraries and we have a really nice high dimensional data set to work with digitally and analyze. And then we can pick variants from that digitally and synthesize them and characterize them. So just the first thing to ask there is the panning working? And what you see is a very nice mutual exclusive enrichment where things are either enriched in the receptor binding domain enrichment, or they're enriched in this competition enrichment, but very few are enriched in both. So that tells you that at least this process is consistent. And if you embed the full repertoire of nanobody sequences in some sort of space where you can visualize it. So this is a sort of a representation of the genetic space of these nanobodies, where 2 neighboring dots, each dot is a distinct nanobody sequence. 2 neighboring dots have very similar nanobody sequences, and you overlay, how enriched they were in various conditions. You can see nice consistent patterns where you see clouds where things were enriched in the same condition. But because these are antibodies, there's an additional layer of complexity in that they emerge from a single cell that was this VDJ recombination event, and then that was stimulated by an antigen and expanded and mutated as it went, and that was caused, contributes to the diversification. So if you group them by their antibody lineages, this is extremely helpful for helping select things, because you expect the functional behavior to be very similar. The potency can change, but the functional behavior should be very similar within a lineage. So taking all of this into consideration, we made a selection of a large number of these and put them through various sort of funnel-based, we screened lots in the beginning and narrowed down. And we wound up with a small set of some extremely interesting nanobodies. So some of them are sort of best in class when it comes to monovalent neutralization potency. So E2 and C7 are those. But those also, like Taiwan, have no breadth. We do have some that are much broader and more potent. So G6 is currently the one that we're paying a lot of attention to. I'll focus on that in detail, but we've also done some more characterization, mapping the epitope, looking at where they're binding, partly to understand how this all interacts with the variant questions, and partly just to get a better handle on their characterization. And so the 2 that are very potent bind in this S2 interface, just like Taiwan does, and that explains why they don't have any breadth. G6 binds to an epitope that's off the side of the receptor binding domain, where there are no mutations in the variants of concern, and actually a lot of conservation between that and SARS-1 and across the sarbecovirus clade. So these are epitopes mapped by something called hydrogen deuterium exchange mass spec. So it's a little bit coarse, but it gives you a reasonable enough answer. And so, like, for example, G6 is just 1 dot in this entire cloud of nanobodies, but the lineage studies show us that this cloud over here at the top are all relatives of G6. So now we can go and look in more detail at different ones and see if we can find some that are more potent than our current candidate G6. And G6 is very potent and very broad. So this is Taiwan, which only neutralized the founder variants, G6 is close to 2 orders of magnitude more potent, but also seems to cross-neutralize beta and delta, and we're working our way down the list, characterizing them. So that's quite promising. We've also found others that behave in very interesting ways. So this is Foo for finding Foo2, binds in this very interesting pattern where there's one binding interface with the receptor binding domain on one side of the nanobody, and a different binding interface on a completely different part of the nanobody that binds to a different, an RBD interface that's from a different RBD. And together, these cause the 2 receptor binding domains to lock in the symmetric configuration with 2 nanobodies in between them. And if you look at what happens in the full spike protein, this causes the 3 receptor binding domains on each spike to flip into this up configuration and locks the 2 spikes together. And we recently got data from negative stain showing that this actually causes SARS-CoV-2 viral particles to aggregate and stick together. So it's a very interesting mechanism of neutralization and quite unexpected from a nanobody. And in challenge models, we've shown that this Foo2, it's less potent than G6, but we isolated it first, so it's sort of ahead in our pipeline. It shows moderate levels of protection against wild type and against the beta variants, and G6 is substantially more potent. So when we get to the G6 characterization, we expect this to perform even better, those are sort of our next ongoing experiments. So next steps for the nanobodies are basically further characterization of G6 in these human ACE2 mouse challenge models. And so far, we've conducted one experiment, and it looks like it works as a treatment against alpha, beta, gamma, delta, and that's ongoing. We've just completed the first experiment and seen the weight loss trajectories. We're also investigating alternative members of these lineages, especially the G6 lineage, and looking at ways of generating multivalent constructs that can maybe enhance their potency even further. And then we're figuring out how to take the next steps and bridge this into something that's practically useful against the pandemic, which is quite new for us. We're much more further away in our positioning to human clinical studies than Teresa is, for example. So basically, that's what we've been up to. I'm extremely grateful to collaborating labs on this. So Gerald McInerney and Leo Hanke in his lab for the nanobody isolation studies. Nila Carlson-Hedestam, especially her student Marco, for the animal immunization studies. Jonathan is a collaborator on all the human ACE2 mouse challenge work. Everyone in my lab who's worked on this, especially Daniel Schuard, who was critical in setting up the neutralization assay, and that's been absolutely essential for the majority of this work. And as Sai mentioned at the beginning, we're funded by a Horizon 2020 program project, and my lab has additional funding from the Swedish Research Council. So with that, thank you very much.